All right, welcome everyone on Zoom. We're going to get started in just a minute. If at any time there's audio problems, please let us know in the chat and we'll try to fix that. Okay, so thank you all for coming. Welcome to Emily Longman's Exit Seminar. So we'd like to extend a special welcome to members of Emily's and Josh's families who are here today. We're thrilled that they're able to join us. It's really wonderful. So my name is Eric Sanford. I'm Emily's PhD advisor. And as is the tradition for exit seminars, I'm going to offer a brief introduction with some thoughts about Emily, and then I'm going to turn it over to Emily to talk to you about her dissertation research. So Emily arrived here in our graduate program with an absolutely stellar record. She earned her bachelor's degree in biology from Brown University in 2015. She graduated magna cum laude with a GPA of 3.97. She then went on to work as a research technician at Northeastern University for 2 years near Boston, where she gained a whole lot of experience in marine ecology working in the lab of Dr. Jeff Trussell. And then Emily arrived here in our graduate program in fall 2017. And for, oops, I have to advance here. Or here. There we go. And for the past 6 years, Emily's been a really central part of all aspects of my lab group. Emily's a real natural leader for my lab group. And as you all know, Emily's made many contributions to the Bodega Marine Lab community, to UC Davis and beyond. So from the time of her arrival in my lab, Emily fit right in. She immediately caught on to the dress code in my lab, as you can see here. Like most people in my lab, Emily is a really big sports fan. She's quite knowledgeable about sports and enjoys watching sports of all kinds. She likes playing sports of all kinds. And like most people in my lab, Emily is very competitive. Here she is winning the limbo contest, much to the amazement of all of her students. And Emily's a really good sport. She doesn't mind competing, even when she has no chance of winning. So, on a more serious note, Emily is one of the most natural field researchers that I've ever met in my very long career. So running experiments out in the intertidal zone requires a very special skill set. You have to get up early in the morning. You have to be comfortable walking over slippery rocks, avoiding giant waves, staying highly organized, and doing lots of manual work really quickly and efficiently before the tide comes back in. And so Emily shines in all of these areas and can single-handedly do the work of at least 3 normal people. So she is an incredible field researcher. Emily's also the fastest walker that I've ever met. And that's coming from someone who used to consider themselves to be, a pretty fast walker. When we realized just how fast Emily walked in the field, we tried to slow her down by putting all this extra weight in her field buckets and kind of weighting her down with extra gear. But this doesn't slow her down at all. So she can carry twice as much gear as anyone else. And despite her small stature, Emily is definitely a unstoppable force of nature in the field. And so, as you'll hear about in just a minute, Emily has completed an amazing dissertation that tackles some of the most pressing unanswered questions at the intersection of evolution and ecology. But I just want to highlight that completing a PhD is really no small challenge. There are always lots of ups and downs, and plenty of obstacles to overcome in any PhD. So in Emily's case, she faced an additional major and unexpected obstacle, which was a global pandemic. And so this added many extra challenges to crossing the finish line, to completing a PhD. When the pandemic hit and essentially shut down the world, it just so happened that Emily was in the midst of experiments that required raising thousands of live snails that had to be fed regularly at the lab. And of course, people couldn't work together in the lab during the early stages of the pandemic. So we essentially turned over our whole lab space to Emily, who worked tirelessly on her own in isolation for months, often seeing no other humans at the Marine Lab. And despite these challenges, as you'll hear, Emily completed a remarkably ambitious set of experiments in the lab and the field, involving hundreds and hundreds of replicates in the lab. This is just a small corner of her kingdom, and a very large and ambitious field experiment in the Rocky Intertidal Zone. During the pandemic, she even managed to recruit a reliable field assistant at that time. And I'll just mention briefly that Emily only has time today to present results from 2 of her dissertation chapters. She completed a whole other big project on historical ecology, which in and of itself is an amazing story. And she also has a large genomics project underway in Rachel Bay's lab on campus. So the amount of work that Emily's accomplished here at UC Davis is really remarkable. So Emily is not one to let challenges stand in her way or dampen her enthusiasm for science. So beyond her research, Emily's been a really outstanding teaching assistant. She helped me teach my field courses here at the Marine Lab during 4 different summers, which was really fun. She's been an exceptional mentor to many undergraduate students at UC Davis, some of who are here today, which is wonderful, and others who I think are on Zoom. Emily has also been a very active member of our BML community. So she served on numerous committees. She's been the president of the Bodega Marine Sciences Association, or BIMSA, here at the lab. She's done a lot of public outreach, like here at the local farmers market. She even organized 2 public film festivals, one in Davis and one here in Bodega Bay, in order to help graduate students communicate their science more broadly to the public, which is very successful. She's been a key member of the Bodega Bat Stars softball team, and just an all-around tremendous team player and a very generous member of our small community here at the Marine Lab. So exit seminars are always a little bit bittersweet. We're all going to be sad to see Emily depart BML at the end of 2023, but we're very excited that Emily was awarded a very prestigious NSF postdoctoral fellowship to work at the University of Vermont with Professor Melissa Pespeni, who some of you met this summer, where Emily's going to be continuing to build new skills in cutting-edge genetics and genomics. So without further ado, I'm happy to turn it over to Dr. Emily Longman.
Okay. Can people hear me? Yeah. Okay. And people can hear me on Zoom? Okay. Thank you, Eric. That was a wonderful introduction, and I haven't seen many of those photos myself. So that was really fun and exciting. And thank you all for coming today. It's so nice to see so many friends and family here, and so many former students doing amazing things, and other graduate students and friends and family. So, and everyone from the community together. So I'm really excited to talk to everyone today about part of my, or a good chunk of my dissertation work, which is titled Drilling Deeper, Exploring Local Adaptation and Eco-Evolutionary Dynamics in a Rocky Shore Community. So a lot of seminal ecological research has been performed at the level of the quadrat or cage. Theories about succession, disturbance, and keystone species have all been developed using these very simple tools. However, it remains unclear whether these studies done at the local level of the quadrat can be extrapolated to predict how processes play out over larger spatial scales and over longer time periods. We know that there's a lot of environmental variation present across large spatial scales that might alter ecological processes. This is a satellite image showing sea surface temperature from June, and you can see that there's quite a bit of variability in sea surface temperature across the Pacific Ocean. There is also a lot of variation in natural ecosystems through time. So as I progress from summer into fall, you can see this warm water anomaly that's been building this fall. Thus, to understand community dynamics, we really need to consider how ecological and evolutionary processes might vary through both space and time. Historically, marine ecologists have neglected the importance of evolution in structuring patterns across scales used in most studies. This is due to the fact that many marine organisms have planktonic larvae, as shown here, and were thought to disperse great distances, leading to high gene flow and population connectivity along the coast. But a growing body of work has shown that many marine species display patterns of restricted connectivity across their range as a result of life history, larval behavior, or oceanographic features leading to the retention of larvae near their natal habitats. This has led to growing awareness of the importance of adaptive variation in marine ecosystems. Given that many species display low gene flow among populations, there's greater potential for natural selection to act on local populations, such as at the level of rocky headlands distributed along the coast, as shown here. And that can lead to patterns such as local adaptation. In addition, if pools of variation exist within populations, there's also the potential that selection might act on this variation over short timescales, leading to rapid evolution. Despite recent attention to these topics, there's a lot that we still don't know about adaptive variation in the ocean. So with this in mind, these are the two main questions that have driven most of my dissertation research, with the first being, what forces generate and maintain adaptive variation within and among marine populations? And second, what are the ecological consequences of this variation? So I've been studying these two questions across two different scales, with the first of which being biogeographic scales, and the second being rapid temporal scales. So for the first half of my talk, I'm going to go into biogeographic scales. The geographic mosaic theory of coevolution is a framework for studying reciprocal selection of co-evolving species across a landscape. This theory is based on the assumptions that species are often collections of genetically distinct populations, that interacting species often differ in their geographic ranges, and lastly, that species interactions often differ among environments in their ecological outcomes. So the kind of different populations are being depicted here as these kind of amorphous blobs. So as a result, across the range of two interacting species, there are regions where coevolution is strong, or hotspots shown here in red, and regions where coevolution is weak, or cold spots, which are shown here in blue, and there's kind of everything in between. I particularly find the cold spots very interesting, as these can occur if the geographic distribution of the two interacting species are not completely aligned, such that one species is absent, or these can occur when both species are present, but additional biotic or abiotic factors alter the strength of the interaction between the two species. Predator-prey species interactions are ideal systems for studying coevolution because the risk of mortality from predation or from insufficient food consumption creates really strong reciprocal selection. And many of these species interactions occur across very large spatial scales with varying abiotic and biotic forces. An example of the geographic mosaic theory of coevolution is in the predator-prey interaction between the Sierra garter snake and their toxic newt prey. So the left figure here is showing the level of toxin levels across the newt's range. The middle panel is showing the level of resistance across the snake's range. And the furthest to the right panel is the level of phenotypic mismatch among prey and predator phenotypes, with hotspots being represented in red and cold spots in blue, where the latter are situations where newt toxicity and snake resistance are poorly matched. Either species in this species interaction can be influenced by environmental gradients. For example, prey abundance, nutritional value, and defenses can all vary spatially as a result of external forces. An example of this is in the commercial clam shown here, which displays variation in calcification rates and growth along 400 kilometers of the Adriatic Sea as a result of variation in solar radiation, sea surface temperatures, salinity, and chlorophyll concentrations. And this variation in calcification can impact their vulnerability to predation. Understanding the adaptive landscape that shapes species interactions also requires considering temporal variation in environmental conditions. This landscape may shift continually through time with biotic or abiotic changes, which can differentially impact the interacting species. And so these changes may shift coevolutionary hotspots to cold spots, such as this red hotspot in the lower left, may switch to a cold spot or vice versa. So these changes could arise via range shifts or contractions, such that predator and prey may no longer interact in some regions, or a third species may shift its range and influence the dynamics of the focal predator or prey. Alternatively, abiotic changes may alter the traits of a given species, which could subsequently modify how the two species interact. And evolved differences in predator traits can in turn drive strong eco-evolutionary effects in the surrounding community through both direct effects and indirect effects on other species. So this gets me to the focal predator and prey species interaction that I'm going to be talking about for most of the rest of my talk. So the predator that I'm going to be focusing on is the channeled dogwalk, Nucella canaliculaata. And I'm going to be mostly focusing in on one of its primary prey species, which is the California mussel, Mytilus californianus, which is shown kind of all around the snail here. And this mussel is a critical foundation species that creates these very large mussel beds along the west coast of North America. These snails consume their prey by drilling a tiny hole through the shell of their prey. They do this using a chemomechanical mode of feeding, in which they alternate between secreting acid from their accessory boring organ and using the radula to scrape away the weakened shell. Nucella canaliculaata lay clusters of benthic egg capsules during the spring and summer. Young snails will develop in these egg capsules and then ultimately hatch out and crawl away, establishing in the surrounding area. Thus, the species has very limited dispersal. Nucella canaliculaata live within mussel beds on wave-exposed rocky shores. So populations are separated along the coast by intervening sandy beaches. And previous research has shown that populations display a pattern of isolation by distance. Previous research by my advisor, Eric Sanford, has documented geographic variation in this predator-prey interaction. Eric did this by raising snails from populations in both California and Oregon in the lab through 2 generations, and then tested their ability to drill mid-sized Mytilus californianus mussels. He was able to show that dogwalks from California, which are on the left-hand side of the dotted line, were readily able to drill these mid-sized mussels, while those from Oregon, which are on the right-hand side of the dotted line, generally could not. So this gets me to the more specific question that I hope to address in this project, which is, how do spatial mosaics of selection drive adaptive differentiation among coastal populations of consumers, specifically Nucella canaliculaata? And my first hypothesis is that Nucella canaliculaata populations differ in the thickness of mussel shells that can be drilled successfully. To answer this question, in 2019, I went to 6 sites, 3 in California and 3 in Oregon, and I collected sets of egg capsules. With each set of egg capsule were laid by a single female, so I classified sets of egg capsules as families of snails, since they all were either siblings or half-siblings. I brought the egg capsules back to BML, where I hatched and reared the snails in the lab on a common diet of thin-shelled Mytilus trocellus mussels. Raising baby snails is a lot of work and is very labor-intensive, and so I couldn't have done this without the amazing help of a former UCD undergrad, Maddie, shown here. So in June of 2020, I then did a very large 25-week-long experiment where I quantified snail phenotype, and I identified the largest and thickest mussel that these snails could drill. And I performed this experiment on 10 adult snails from 8 families from each of the 6 populations. To do this, I took advantage of the fact that larger Mytilus californianus mussels have thicker shells, as shown in the figure on the top left. So I challenged individual snails with a series of mussels of increasing size. I started all the snails at the smallest size class. Sorry, thing just popped up. Just move this. I started all the snails at the smallest size class, and then if they were able to drill one of those mussels, they were subsequently offered thicker mussels of the next size class. And of course, that messes up. Sorry. Okay, so the mussels from the first 6 size classes were all from Bodega Marine Reserve. And if snails were able to progress past the 150 millimeter mussel from Bodega Marine Reserve, they were subsequently offered a 150 and then a 170 millimeter mussel from Strawberry Hill, Oregon. And based on preliminary data, we had assumed that these mussels from Strawberry Hill in Oregon would be exceptionally thick and would represent the end point of our continuum. That's fine. Okay, so I ultimately analyzed the largest mussels drilled for each individual snail using linear mixed effects models. And so to kind of orient you to this graph, I have population along the x-axis. I'm going from the southernmost population on the left to the northernmost populations along the right. And I have the maximum length of mussels drilled along the y-axis. So when I add in the data, you can see that dogwalk populations were strikingly divergent in the maximum size of mussels that these snails could drill successfully. Dogwalks from California were capable of drilling much larger mussels than those from Oregon. The Oregon snails were only able to drill mussels that were typically less than 5 centimeters in length, while some of the California populations were capable of drilling mussels that approach 15 centimeters, which are some of the largest mussels that you can find in these mussel beds. So then to identify the maximum drill hole depth, I took the largest several mussels drilled by each individual snail, and I cut them on a bandsaw through the drill holes or partial drill holes. And I did this because mussel thickness varies along the anterior to posterior axis along the mussel. So the largest mussel drill didn't always contain the deepest drill hole. And so I took photographs of these drill holes or the partial drill holes by placing these cross sections flat on a scanner. And then I measured the depth of the drill hole using image analysis software. So similar to the results on maximum length, maximum drill hole depth differed significantly among the populations. So this graph has the populations along the x-axis, but maximum drill hole depth along the y-axis. And you can see that the dogwalks from California had the capacity to drill holes that were 4.4 times deeper than those from Oregon. So going back to my main question for this project, I hope I've been able to prove to you that the drilling capacity of populations of Nucella canaliculaata differ greatly across the West Coast. However, this doesn't answer what has caused this pattern. So my second hypothesis is that Nucella canaliculaata populations are adapted to a consistent coastal mosaic of mussel shell thickness. The California Current is an eastern boundary upwelling system that during certain times of the year, upwelling brings cold, nutrient-rich water from the deep ocean to the surface. In addition, upwelling creates a generally persistent spatial mosaic of acidified waters in nearshore habitats. So this figure shows the level of pH severity for 3 years across much of California and Oregon. Oceanographic conditions can in turn influence patterns of mussel growth and shell thickness. And if this spatial pattern of upwelling and pH has been persistent over long timescales, this may have created a spatial mosaic of mussel shell traits along the West Coast with effects on the evolution of the drilling ability of Nucella canaliculaata. So I was interested in analyzing shell thickness across the same 6 populations that I studied snail phenotype. And lucky for me, Eric has saved lots of boxes of mussel shells from his previous research. So I was able to analyze mussel shell thickness across 2 decades, with the first set of shells being collected in 2000 to 2001, the second set being collected in 2008 to 2009, and then I collected mussel shells in 2019. So I cut all of these mussels, or the left valve of all of these mussels, at 1/3 their length from the anterior end on a bandsaw. And I chose this region since this is the most commonly drilled region by Nucella canaliculaata. So then I scanned these cross sections, and using photos, I then was able to calculate average thickness via imaging software. So I ultimately analyzed the log of the ratio of mean mussel shell thickness at 1/3 the length of the mussel to mussel length. And using this ratio allowed to control for differences in the length of mussels sampled. So to orient you to this graph, I still have the same populations along x-axis with the California populations on the left and the Oregon populations on the right. And this is a lot of data, so I'm going to walk through this slowly, starting with the first time period and then subsequently adding the later time periods. So in 2000 to 2001, the two Central Oregon sites had the thickest mussels, while the Northern California site of Van Dam had the thinnest mussels. A very similar pattern was observed in 2008 to 2009. And then in 2019, the spatial pattern had changed, where the thickest mussels were still found at Strawberry Hill in Oregon, depicted here as SH, while the thinnest mussels occurred at Fogarty Creek and Van Dam. And when you analyze each site separately, there are clear temporal patterns of mussel thinning over time, where mussel shells at all of the sites except for Soberanes Point were thinner in 2019 than in 2008 to 2009. And the two sites that had the greatest amount of thinning were the two Central Oregon sites, which previously had the thickest mussels. So this finding is in line with previous work from both California and Washington, where researchers have compared the mineralogical composition of archival shells with modern day counterparts and have been able to show that ocean acidification associated with climate change is altering mussel shell growth and calcification. But the main question from this project is how these two pieces fit together of drilling capacity and mussel shell thickness. So this figure is showing the estimates of drilling capacity along the y-axis and shell thickness along the x-axis for each of the 6 sites for the 3 time periods, with the 6 points in each time period corresponding to the 6 sites. And what you can see is that for the first 2 time periods of 2000 to 2001 and 2008 to 2009, there's a negative relationship between snail drilling capacity and mussel shell thickness. However, this pattern breaks down in 2019. So these results suggest that the spatial variation in shell thickness has contributed to adaptive differentiation among populations of dog whelks, where there's a spatial mosaic of shell thickness, with shells consistently being thinner in California than in Oregon over time. And thinner shells in California is associated with greater drilling capacity in dog whelk populations, a pattern which on the surface may seem counterintuitive. However, we hypothesize that these thinner shells in California may have initially led Nucella canaliculaata in California to target Middleus californianus as a viable prey item. And then over time, selection to prey on larger mussels with greater tissue and thicker shells favored the continued evolution of greater drilling capacity in these snails in California. In contrast, we hypothesize that the thicker Middleus californianus shells in Oregon dissuaded dog whelks in this region from preying on this species. In addition, previous research has shown that the abundance of alternate prey, specifically barnacles and thin-shelled Middleus trochelus mussels, is much greater in Oregon than in California. And this might have further weakened the selection pressure on Oregon dog whelk populations. So our findings are consistent with this hypothesis that the strength of selection varies with prey defenses across the overlapping ranges of these two species, with strong prey-driven selection on the predator in California and a coevolutionary cold spot on the Central Oregon coast. In addition, we believe that there are ecological consequences for these divergent phenotypes, such that this intertidal predator may serve different functional roles across its biogeographic range. We hypothesize that Nucella canaliculaata may have greater community effects in California than in Oregon due to the stronger impacts on the foundation species Middleus californianus and their ability to drill these very large mussels. While in Oregon, Nucella canaliculaata may have a smaller influence on rocky shore community dynamics, and this is in line with a lot of previous research on the Central Oregon coast, which has shown that dog whelks have much weaker effects than Pysester ocraceus, the ochre sea star, which is a keystone predator on rocky shores and is known to dictate mussel bed dynamics. However, the selection landscape appears to be changing as a result of ocean acidification. And this may impact prey-driven selection on populations of the dog whelk Nucella canaliculaata. However, the capacity and rate at which Oregon populations of Nucella calyculata might evolve to target Mittalis californianus will depend on the level of standing genetic variation present in these populations. So a greater understanding of gene flow in this species and population structure along the coast is needed to predict how this species may or may not adapt as conditions change. So stepping back a little bit, having a solid understanding of species life history and dispersal potential is critical when studying interacting species across a landscape, because mismatches in dispersal can result in unique selection mosaics for interacting species with consequences for understanding local adaptation and ecoevolutionary dynamics. For example, a weevil, shown here, that preys on the seeds of the Japanese Camellia has much greater dispersal potential than its host plant. As a result of this, the weevil has led to a homogenizing effect that promotes the evolutionary convergence among populations of the host plant. In contrast, in our study system, the prey species, Mittalis californianus, has really high dispersal and high gene flow across its geographic range, whereas the predator, Nucella calyculata, lacks planktonic dispersal and is highly differentiated among sites. These differences in dispersal potential and the asymmetric strength of selection pressures on predator and prey may lead to a system where spatial variation in prey traits, specifically muscle shell thickness, are environmentally driven, while low dispersal in the predator promotes local adaptation and consumer phenotypes with much broader ecological consequences in the surrounding community. So going back to our ecological theory, I want to highlight again the importance of studying species interactions within a geographic context of varying environmental pressures and biotic forces. These forces likely play an underappreciated role in dictating evolutionary patterns of phenotypic divergence with ecological consequences for communities, especially in a rapidly changing world. So I want to summarize what we've covered so far. I hope I've been able to demonstrate to you that there's a striking mosaic of drilling capacity in the marine snail Nucella calyculata, and that this predator phenotype is associated with a variation in muscle shell thickness. However, this selection mosaic appears to be changing as a result of ocean acidification associated with climate change. So far, we've discussed the mechanisms and consequences of adaptive variation across large spatial scales and decadal temporal scales. So now we're going to switch scales here and begin to think about these questions over rapid temporal scales. So there's increasing awareness that evolution can proceed rapidly, with genetic changes occurring rapid enough to have measurable impact on simultaneous ecological change. For example, previous research on Darwin's finches in the Galapagos Islands has established that periods of drought can drive changes in plant communities and therefore seed availability, which can drive the evolution of beak morphology in finches, as well as alter the population growth rates for species with different beak morphologies. Increased recognition that evolution can proceed rapidly has resulted in the emerging field of ecoevolutionary dynamics. This field analyzes the reciprocal impacts that ecological and evolutionary processes can have on one another on contemporary timescales. Studies in this field so far have shown that ecoevolutionary dynamics can influence species diversity, predator-prey cycles, community structure, and the success of biological invasions. And although this field is getting a lot of attention right now, there are still, I think, a lot of gaps in our knowledge. I think the biggest unanswered question is if these feedbacks are important or trivial in nature. Most of the experiments have been performed in lab settings or in mesocosms. These experiments are a really important first step for studying ecoevolutionary dynamics. However, field experiments are needed to determine whether ecoevolutionary feedbacks are swamped or amplified by external factors in more complex natural environments. Second, many of the ecoevolutionary dynamic studies so far have generally focused on pairs of species and have seldom tested for their effects at the community or ecosystem level. Also, most of the experimental studies on eco-evolutionary dynamics have generally compared 2 or more locally adapted populations that have evolved under spatially divergent selection. For example, researchers have taken guppies from different rivers in Trinidad that have evolved under different predation regimes, as shown in the figure here, and then tested them in mesocosms at one location. And while such studies demonstrate that evolutionary processes can influence ecological dynamics, this approach often relies on substantial phenotypic variation that has evolved through divergent selection imposed on separate populations over decades to a century or more. In contrast, there have been increasing calls for studying the dynamics of systems where there is no separation in time between the evolutionary and ecological processes under consideration. So one promising approach for studying eco-evolutionary dynamics is to explore whether abiotic and biotic variation acting over rapid timescales from months to years might impose selection on existing within population variation and functional traits, especially those that have links to community dynamics. And I think the Nusala Canalicculata system seems to be this ideal system for testing the community level consequences of eco-evolutionary dynamics. And that is because this is a predator that exhibits a substantial variation in foraging traits and drilling ability, has a heritable basis. So in this figure of Eric's, which I showed previously, I want to focus in on the Bodega Marine Reserve population, which is highlighted here, which contains a mix of drilling phenotypes that varies both within and among Nusala Canalicculata families. These snails also have a relatively fast generation time and have really high early life mortality, as high as 90 to 99% during the first 2 months of life. So upon hatching, these juvenile dogwalks are really small and are dependent on a supply of newly recruited prey that are spatially and temporally variable in their abundance. In addition, Bodega Marine Reserve is located at a region along the coast that is known to have high interannual variation in recruitment of both barnacles and mussels, as well as have a lot of variation in upwelling with high variability in pH between years, which could result in temporal variation in mussel shell thickness. So due to the substantial environmental variation at Bodega Marine Reserve and the high early life mortality in Nusala, there's the potential for strong selection to occur within each generation of snails. Previous work has shown that Nusala can alter community processes, specifically mussel bed succession. So winter storms and large waves frequently dislodge mussels and create disturbance gaps that then go through this well-documented sequence of succession, going from a bare space to a mix of species and then ultimately back to being dominated by mussels. And this whole process from bare space to mussel beds can take approximately 2 to 4 years. So this gets me to the focal question for this project, which is how does temporal variation in selection drive eco-evolutionary dynamics in an intertidal predator-prey interaction, and what are the cascading community effects? So I hypothesize that temporal variation in prey could impose strong selection on within population variation in drilling phenotypes in Nusala Canalicculata, and subsequently that variation in drilling phenotypes could impact mussel bed succession. To test this hypothesis, I conducted a selection experiment where I reared newly hatched dogwalks on different prey treatments, mimicking a situation in nature where cohorts of snails would hatch out and encounter different prey communities. So in summer of 2020, I collected 18 sets of Nusala Canalicculata egg capsules from Bodega Marine Reserve and 4 sets of egg capsules from Soberanes Point, depicted here as SBR. And similar to the last project, I'm going to refer to egg capsules that are from the same cluster as families of snails. So the population of snails from Soberanes Point were used as a control in this experiment, since previous research had shown that after 2 generations on a common laboratory diet, this population consisted of individuals with uniformly strong drilling capacity and little variation among individuals. So dog whelks were hatched at BML, and snails from each family were split among 4 different diet treatments for a 3-month-long selection phase. The diets were a controlled diet of thin-shelled Mitilus trochelus mussels, which is what I used during the first experiment, during the first half of my talk. I then had 2 diets of Mitilus californianus coming from 2 different populations. And then my final diet treatment was a diet of acorn barnacles. The 2 Mitilus californianus treatments were used to represent differences in shell thickness, since previous research had shown that adult Mitilus californianus from Bodega Marine Reserve had thicker shells than those from Soberanes Point. And I just want to note that the color scheme being used here to represent these 4 diets is going to be used for the rest of my talk. So again, raising snails is a lot of work. So I just want to point out a few people who were really instrumental to making this project happen. So after the 3 months on the diet treatments, I then assessed mortality for each family. And I analyzed this data with generalized linear mixed models. So I'm going to walk through this figure slowly since it's both a lot of data and I have similar figures set up to this later on. So I have the 4 diet treatments along the x-axis and the percent mortality at the level of the family along the y-axis. So each diet treatment is going to have 2 bars, with the left bar, which is going to be darker in color, is going to represent the snails from Bodega Marine Reserve. And the right bar, which is lighter in color, is going to represent the snails from Soberanes Point. So when I add in all of the data here, you can see that mortality of dog whelks varied among the diet treatments, with snails raised on Mitilus trochelus having the highest survival, and the snails raised on the thick-shelled BMR Mitilus californianus mussels having the greatest mortality. And I also want to note that the snails from the 2 different populations, so the 2 bars for each diet treatment, did not differ. So after the 3-month selection phase, I then reared all of the snails out on a common diet of Mitilus trochelus until they were adults. At which point in time, I ran a laboratory scoring experiment to quantify the snail's drilling capacity. To quantify dog whelk phenotype, I tested individual adult Nucella canaliculatta on their ability to drill mid-sized Mitilus californianus over the course of a 100-day experiment. So I gave each individual snail 2 5 to 7 centimeter long mussels and checked these containers every 3 weeks. I assessed drilling capacity both as a binary response of if a snail was able to drill at least 1 mussel over the course of this 15 weeks. And I also counted the total number of mussels drilled per snail. Due to the nested structure of the data and the high level of variation present within or between populations, I ran Bayesian hierarchical models to analyze the data. And for the sake of time, I'm only going to go over the binary drilling data since the data on total number of mussels drilled was very similar. So similar to the previous graph, I have the diet treatments along the x-axis. And along the y-axis, I'm showing the proportion of snails for each family that were able to drill at least 1 mussel during the scoring experiment, but the stats were run on individual level data. And what you can see is that dog whelks from Soberanes Point, which are the white bars for each diet treatment, had much higher frequency of successful drilling than those from Bodega Marine Reserve, with all of the snails from Soberanes Point being able to drill at least 1 mussel. So early life diet did not modify the drilling ability of dog whelks from Soberanes Point. In contrast, early life diet had substantial effects on the percentage of surviving Bodega Marine Reserve snails that were strong drillers. Drilling success was highest for those Bodega Marine Reserve snails that were raised on the BMR Mitilus californianus treatment, which are shown here in dark red. And as a reminder, these were the snails that had the highest early life mortality. So far, I've been able to show that early life diet results in differential mortality that affected the drilling capacity of adult dog whelks. The second piece of this is now closing this loop and testing the ecological consequences of these divergent phenotypes. So to test this, I did a field outplan experiment where I assessed whether variation in drilling phenotypes would have ecological consequences on mussel bed succession. I predicted that dog whelks would slow succession compared to plots where dog whelks were excluded. And second, I predicted that dog walks with a greater proportion of strong drilling phenotypes, which was shown to be the snails based on the BMR Mittalis californianus treatment in our lab experiment, which drill more and larger muscles and slow the progression of succession. So to do this, in May of 2020, I cleared 16 mid-intertidal sections at Muscle Point. I then let these communities develop naturally for a year until the plots contained a mid-successional community. At which point in time, in May of 2021, I installed stainless steel mesh cages with removable lids into these 16 regions. The cages were in a block design where 5 cages were placed within each of the 16 larger regions, where each block represented snails from the same family, with each cage randomly being assigned to contain snails from one of the 4 diet treatments. Plus, there was 1 reference cage that contained no dog walks. So if I take off the lid on one of these cages, you can see that the plots all started at this mid-successional community where there was this mix of acorn barnacles, gooseneck barnacles, and a few small mussels. And I chose this stage to start my experiment because I thought this would be the point in time that the snails could have the largest impact on the trajectory of mussel bed succession. So in July of 2021, I placed 5 adult dog walks from each of the 16 Bodega Marine Reserve family by diet treatments into each cage. And then every 8 weeks for the next year, I would go out, I would remove the lids, photograph the plots to assess percent cover of sessile species. So the progression of just 1 cage through time is being shown here. So you can see succession happening over the course of the year. For the percent cover analyses, I grouped species into 7 functional groups, which were bear rock, acorn barnacles, gooseneck barnacles, mussels, other sessile animals, coralline algae, and a final group of algae and surfgrass. And I just want to point out 2 former UCD undergrads, Anna and Jackie, who are really helpful, given that this was such a big project. So I analyzed the percent covered data using non-metric multidimensional scaling ordinations in Permanova. I visualized the NMDS graphs for each experimental check, with this figure being shown here, being the community composition right before I outplanted the snails. So each dot represents the community composition based on the 7 functional groups are present within a given cage. And the color of that dot represents the treatment, with gray being the reference cages where no dog walks were added, and the 4 colors represent the early life diet treatments of the snails placed within those cages. And the ellipses are the 95% confidence ellipses. So this ordination, as this was right before I put out the snails, it illustrates that the community composition did not differ across the experimental cages at the start of the experiment, given that everything's kind of on top of each other. So these are the ordination plots through time. And you can see that over time, all of the dots and kind of ellipses move towards the mussel section of those figures, which highlights the progression of mussel bed succession. And over the course of the year, the trajectory of succession diverged most clearly for the reference cages where there were no whelks added, which are the gray dots and gray ellipses. But the big question that I was testing is how the colored ellipses differ. And going back to our predictions, we would predict that the red ellipses that represents the snails based on the BMR Mittalis californianus, which had the highest early life mortality and diverged most strongly in drilling phenotypes when measured in the lab, would slow succession. And although there were some community differences, there were substantial overlap and there was no clear differences in the trajectories of mussel bed succession for those 4 treatments where the dog walks were added. So to identify differences among treatments, I also ran follow-up univariate models on the 7 functional groups. To keep things brief, I'm only showing the results from the mussels shown here, since the mussels are the end stage of succession. So I have time along the x-axis and the percent cover of mussels along the y-axis. And what you can see is that the reference cage shown in gray, where the dog walks were excluded, differed primarily by containing many more mussels than the 4 treatments with dog walks. And again, the key point is that the red diet treatment representing those snails based on the BMR Middleus californianus did not differ in muscle cover, contrary to our prediction. And then also at the end of the experiment, I went out and I collected all of the organisms within the cages and counted and measured all of the muscles. So the number of muscles within those cages is being shown here. And you can see that the number of live muscles held within the cages differed among the treatments, but only the reference cage where no dog walks were added contained more muscles. The 4 treatments where the dog walks were added did not differ in terms of the number of muscles in these cages. So jumping back to the question of if there are eco-evolutionary dynamics in the system, despite us finding differential mortality among the diet treatments suggesting the potential for selection, and the fact that we saw differences in adult drilling capacity in the lab, eco-evolutionary feedbacks were relatively weak when tested in a natural field setting. And I don't think this is an artifact of just our system. I think there are a number of factors that may have driven this finding, and I think these factors likely apply much more broadly to other systems. So I think eco-evolutionary feedbacks are often swamped in the field by the overriding effects of substantial spatial variation in environmental conditions. On rocky shores, fine-scale spatial patterns, such as recruitment and wave exposure, often vary over very small spatial scales and may impact the rates of succession. So against this backdrop of substantial variation in environmental drivers, the influence of within population variation in drilling traits on succession may be comparatively minor. Second, given that our focus for this project was to have no separation in time between ecological and evolutionary processes, we focused on the variation present within one population. However, there was likely a limited range of within population variation, especially when compared to that found across populations. For example, going back to the figure of Eric's Common Garden experiment, the focal population of Bodega Marine Reserve does include this mix of snails with stronger and weaker drilling phenotypes. But the range of phenotypic variation is less extreme within this population than it is found across the geographic range of this species. And thus far, many of the experiments that test eco-evolutionary dynamics have compared the effects of locally adapted populations or divergent ecotypes. A similar approach has been taken in the field of plant community genetics, where plants have been collected all the way over here from distant diverged populations, and then their effects on insect communities are quantified in a common garden experiment, so a much smaller spatial scale. And I think these approaches often create a mismatch in the spatial scales used to test the relative importance of genetic versus environmental influences on community processes. And lastly, eco-evolutionary feedbacks might be weaker than expected in nature if selection on a population is not strong enough to produce extreme divergence in functional traits. The existence of wild populations comprised of phenotypically diverse individuals suggests that past selection has not been strong enough to eliminate variation in traits under more realistic selection regimes. So even when there's substantial selection, like the selection that we performed in the experiment here, they're likely resulted in a mix of some strong drillers being present in all of the diet treatments. And I think the maintenance of this variability likely dampens eco-evolutionary effects in this community. And I think that likely applies much more broadly to other natural systems. So circling back to our main question for this section, despite evidence that our early life diet treatments generated divergence in drilling traits, the effects of these feedbacks on the community dynamics in the field were relatively weak. And we think that this likely applies more broadly, where a range of factors and constraints likely dampen the strength of eco-evolutionary dynamics in natural communities. So I just want to take the time to summarize what we've covered today. So the first half of my talk discussed how there is striking spatial variation in a predator trait that is correlated with a prey trait that seems to vary along an environmental gradient. The second half of my talk covered how selection on early life diet shapes dog walk drilling capacity, but when tested in the field, these phenotypes do not impact the trajectory of muscle bed succession, suggesting that eco-evolutionary feedbacks may sometimes be dampened in natural communities. And more broadly, and kind of more generally, I think my dissertation highlights the importance of studying species interactions in a broad ecological and evolutionary context, especially when thinking of natural communities in this era of climate change. So with that, I'll take this is my shorter acknowledgements. There will be longer acknowledgements later, but I'd like to thank a lot of people who helped in the lab in the field, as well as my dissertation committee and my funding resources. And then we can take questions now.
Yes, so Emily would be happy to take questions. If you're on the Zoom seminar, feel free to type any questions in the Q&A and we'll read those here in the lecture hall. But for now, Emily will start with any questions here in the lecture hall.
Priya.
Dr. Longman, that was very truly having seen you work through the years, it's incredible to see all of this. I was so intrigued by your findings on your talk where you see ossification connecting on the shell thickness of the muscles. I was curious, are you seeing more happening on the snails themselves? Especially like a difference between Oregon and California, like you saw.
Yeah. I'm going to repeat the question for those on Zoom. So Priya asked this question focusing in on the first half of my talk about the impacts of ocean acidification on muscle. Well, she was intrigued by the fact that ocean acidification was thinning muscle shells, but was curious if that impacted the snails themselves. I think that is a great thing to study. I have not actually studied it. I've been very interested in wanting to study kind of the snails' morphology and shape in greater detail. There's a lot of things I've noticed across this really big spatial scale. I could open a container and tell you which population it was from, even though it's very subtle, but there's like very tiny differences that looked really different. And Eric does have snails from his previous stuff. They feel all stinky, but I would be interested in testing that actually at some point, and that may be possible to actually look at the kind of mineralogical composition or shell thickness of the snails themselves, but I haven't done that. That would be cool.
Very cool.
Anya.
I have a question from your last chapter and you presented, and thinking about the differences in your selected families and groups. Do you think, so I have two parts to this question. One, do you think their preferences for prey changed based on that experiment, or what are the preferences of the snails? And do you think when in the field, they're selecting other prey items besides the mussels that are sunken in those spots, and so maybe competitive interactions are also different? So it's like other mechanisms that are leading to the same result.
Yeah, okay, I'm gonna try to repeat the question for those on Zoom. That was kind of a longer. So you were asking if in the second half of the talk for eco-evolutionary dynamics piece, if raising the snails on the different diets would select for things. So it's or like have other effects on the snails in terms of, I'm assuming you're thinking of like learning and that sort of stuff, or plastic things, or?
What are the preferred, what's the preferred?
Oh, what's the preferred diet?
Bigger shell versus a thinner shell or barnacle?
That's a great question, because someone who's sitting right in front of you did an experiment on that. So Jenna actually did that and tried to, Jenna Kwan, for those who are on Zoom and can't see her, who is a former UCD undergrad and Kendra Chan fellow, did a project actually seeing if these snails from more the first half of the talk, the snails from the different populations, if they both had a preference for the size of the mussels. And it seemed like The ones that were good drillers would go after anything very quickly, and the ones that were poor drillers took a long time to find the mussels that they could drill. And so that was really interesting. And she also did an experiment where she gave snails mussels that varied in terms of the shell thickness. So she gave them a thicker and a thinner mussel, and they couldn't tell the difference at all. They just. So I haven't done that specifically. I think it would depend across this like geographic range. I think like in Oregon, the snails definitely probably prefer chaucelus and barnacles. I think, and those ones seem to crawl around a lot more in general. I think a lot of the ones that are Sean drillers just start drilling anything. And many of the ones in California, most of the California populations, these snails live deep within the mussel bed for the most part. So you have to kind of like break up the mussel bed to get to them. So a lot of the time they're like deep down, and that's mostly where a lot of the mussels are, where mussels are. The barnacles are usually more on the surface. And so that's where like this bodega population seems to be interesting, where there is a mix of phenotypes and there's a lot of variation across years in terms of recruitment of barnacles and mussels. Like this year was a really massive barnacle recruitment year. And I can tell you anecdotally, because I sadly wasn't taking any data, the snail populations are really crazy. And there were tons of them out there. And I think they were taking advantage of the fact that there are lots of barnacles out, because they were like, this is really easy prey. And I think sometimes can be also easier for small ones, for small snails to be able to get through, since barnacles have in general weaker shells than mussels. But a lot of barnacle recruitment can be somewhat variable across space. So somewhat answers your question. Yeah.
There is
a question on, yes.
Want to read that out?
Yes. Given that one of my committee members is on Zoom, I will read Jay's question out loud. So seems like all snails can drill small mussels, and variation is in the ability to drill large mussels. So would you predict that snail phenotypic variation would not affect early life or early succession when mussels are all small, but might have effects later in succession if some small mussels escape predation and become larger? So do you think there would be feedbacks if you ran the experiment longer? And then, then he says some other. Okay, so Jay's question. I think that's a great question. I actually think somewhat the reverse. So I would say by the end, most of my plots were 100% mussels, and there were some of them that like you opened up and the entire cage from the very bottom to the top was all mussels. And I think 5 snails in there almost like can't do enough at that point. That probably would have had to been like a higher density if I was doing it more, but I went out and actually surveyed things. And so I put in what was a natural density at those or at Mussel Point. And so I wanted to do something that was realistic and not artificially inflating it. I think actually there may have been a chance that the snails could have had a bigger impact if I actually did it a little earlier or at a different location. I think Mussel Point is, I mean, many of you have gone out there with me. Mussel Point is very wave exposed. And I think recruitment is very high. And because of the amount of waves, I think succession goes really quickly and things grow really fast. And so I think somewhat the weight of that just kind of overwhelms the impact that the snails could somewhat have in terms of the difference. So it may have been possible if it was somewhere else or maybe earlier, or as I showed that they might not have had any impact in terms of within population variation. I can do another one here, or Sam.
Jay stole my question, but congratulations, Emily. It's really awesome to see all of your hard work up together. But I'm wondering whether, like, I'm curious about how realistic you think some of those questions that looked at more spatial skills and much feedbacks there. And so I'm curious if you have any ideas, like, how much you'd expect long dispersal to play a role, and if you were able to like convince CDFW to like transplant Oregon snails to California, what do you think you would see feedbacks that you think more likely to see feedbacks there?
Okay, I'll repeat the question, for those on Zoom. So Sam's question originally was the same as Jay's, but then he kind of switched the question, to being, the fact that a lot of the eco-evolutionary dynamic studies have usually taken locally adapted populations that are, from further away populations, and then tested them. So I would say in some ways, actually, I think Eric's previous reciprocal transplant experiment in some cases kind of does some of that, where you're taking populations that you did raise in a common lab environment, and then tested the effects at different populations, or tested them here, and then the effects of these populations both in California and in Oregon, and in some cases, I think even just like that reciprocal transplant already gets at some of that. And I think that could be interesting to more look at that study, or kind of look at that study in conjunction with what I did here to think about it in an eco-evolutionary dynamics framework. But personally, I think like the big thing that really is lacking in a lot of eco-evolutionary dynamics literature in general is actually looking at kind of more natural systems and not artificially inflating the amount of genetic variation by kind of taking populations that you already know are different because people have looked at them from the scale of local adaptation. So that's why I thought this was a really great system, where Eric's work had shown that there's a pattern of local adaptation present, and that the Bodega Marine Reserve population contained this already mix of phenotypes that it may be possible to kind of shift through differential mortality.
Yeah, let's just do quick answers to the two Zoom questions. And
Okay.
then we can move on with your-
Okay, so Dr. Jason Sadowski has a question. In the second half of the talk, do you know how many snails would be required to create the sort of successional change you are expecting? Okay, so I got at some of this before. Is it possible only 1 or 2 snails in the outplant would be required to dampen out the changes? So I think this is kind of somewhat two-part. So I kind of answered part of this before. Since I was trying to go after what was like natural densities, I was putting out very similar numbers of snails as what normally exists out there. And I don't know, I think maybe his question is more getting at if just the impacts of the presence of the snails would reduce the amount of muscle cover. And I think it may be possible, although it would depend then on if you're only putting out like 2 snails, I think there's a greater chance that you may have gotten kind of even in the diet treatments that I would have thought would be weaker drillers, you could have gotten randomly one of those strong drillers in there. And so when you have like smaller numbers, you could have like a greater chance that things could be kind of impacted by just small numbers. And the fact that these guys seem to have variation, and I couldn't perfectly select for strong versus weak drillers perfectly in my lab experiment. And then Gina on the chat asked if I looked at gradular morphology. I did not, but I think that has always been something that I would love to look at. I'm actually really interested in trying to dig into in general what is causing these differences in terms of the pattern of local adaptation. And although I think actually looking directly at gradular morphology is going to be a little bit hard, or same with the morphology of the accessory boring organ. I'm hoping to kind of get at that in my postdoc, which is going to be expanding on some of this work, but looking at from a genomic perspective. And I'm hoping to do kind of gene ontology work, which could potentially to identify what is the cause of the better drillers versus weaker drillers. And if it is some sort of acid related kind of thing versus something that's a morphological thing. So I hope to get at that later.
Yeah, sure. Talk about the questions. You have an opportunity to ask additional questions. Talking with Emily later at the reception. But for now, I think Emily is
yes. Okay. So now for the really fun thing. Okay, the real acknowledgements. Okay. So real acknowledgements, I'll start. Many of these pretty photos are Gabe's that he sent me. So gotta appreciate that. But I'll really start with Eric. Eric's been an amazing advisor, and shockingly, I used a lot of the same photos that you did, but flipped around. We have you here hiding in Gabe's fear wall. So Eric's been an amazing advisor. He's also an amazing naturalist, and he inspires so much curiosity about the natural world. Every time I go out in the field with him, I think I learn more things about invertebrates or rocky shores. And I just appreciate all of those times so much. I also have appreciated being able to be your TA for many classes, the summer classes particularly. They're a lot of work, but I learned so much about kind of mentoring, and I learned so many new things every year from the students for their new projects, which is really fun. You're also an amazing writer, and I've learned a lot from that, and my writing has definitely improved drastically because of your comments. And so I'm gonna definitely miss many aspects of being in your lab, from all of like the field excursions and building kind of elaborate setups and that sort of a thing. But I will not miss losing at cornhole. Although some of you may lose at cornhole to Eric and Jackie later this afternoon. I want to thank my dissertation committee. Jay has been incredibly helpful during my entire dissertation since my first year in Davis. And he's made me think much more broadly about community ecology and putting kind of my work in a kind of much broader setting. And especially in my first year in Davis, given that Eric's lab is based out here, Jay welcomed me to come to his lab meetings and things, and I really greatly appreciated that. Rachel, who is my other dissertation committee member, was so generous to open up her lab to me so that I could learn a lot of molecular and genomic skills. And even though that project didn't get into my dissertation because it took me 2 years to sequence the snail, I learned so much working with both Rachel and Brenda, her lab manager, and a lot of those skills are going to be really helpful for me in my postdoc, given that I'm going to be doing a lot more genomic work. So much of my dissertation was completed during a global pandemic. And this could have resulted in a major disruption. But since I already had a lot of experiments going, I was able to kind of chug away at raising thousands of snails, which was really, really great, but also really lonely sometimes. So I want to take this point in time to thank especially a lot of the BML and BMR staff for keeping everything running. During the time of the pandemic, it made my day to see Joe or Philip or someone from down the hall and just wave at them. So I really greatly appreciate that. And I think a lot of, or everyone at BML and BMR are just so amazing and keep this place running during a pandemic, during the big storms, and just even day to day, from everyone to physical plant to ARG, to the reserve staff, to the library. Everyone just kind of makes this place feel like home. I want to thank the Sanford Lab. We're pretty small, but we're a mighty group. Sam and Kira, you are gotten to be two of my closest friends during grad school. And I really appreciate all of you guys for both talking about science all the time, helping me in the field, going out and doing fun activities in the evenings or on the weekends and that sort of a thing. And so I always just appreciate that so much. I want to also thank two other Sanford Lab members, Sarah Marola and Manny. You guys are amazing and have also great friends and have helped me so much during my time here. And I just appreciate it so much. My research involved raising thousands of snails and performing these really massive field experiments and going up and down the coast a lot. And I couldn't have completed it without the help of a ton of amazing people, many of which are undergrads, some are grad students or friends or other things. I think I got everyone on here in one picture. It's really hard. Jackie's selfies really helped for the breakdown of the experiment. That's for sure. But I really appreciate all of you guys for helping so much. It was really fun always going out, and I hope you guys had as much fun doing a lot of this fieldwork as I did. I want to thank my friends, both friends from afar and friends here. I have a lot of great friends that I grew up with and really close friends. And even though I don't get to see you guys as much, I really appreciate it. I really want to thank my BML kind of family. We're a really small community and you guys have been like a home for me for the past several years. if I'm doing big kind of fun activities like Bat Stars or trivia, but a lot of you have become some of my kind of best friends, and I just appreciate it so much. Doing all the kind of fun activities outside. And specifically, I'll note again, I want to thank Gabe for all the stats checks. There's a picture that Josh had taken of us working on stats, staying up until midnight. You're one of the only people I know that I can go to a bar to and talk about statistics for hours on end, and scream at you about stats. And I really appreciate that because I like talking stats. I want to thank my family. You guys have always been incredibly supportive of me. And despite being the only one in my immediate family that has gone into science, you've always been there for me and encouraged me to do what I want to do. And even when I kind of picked up and moved cross country for a dissertation, you guys have always been really supportive. And even though I haven't been able to see you as much, especially during the pandemic, you always are willing to talk on the phone with me in my phone booth of a car for kind of like hours on end while I'm driving back and forth between field sites. And I really appreciate that. And this is where you'll see it's great that we got married because we have lots of pictures. I want to thank the Foss-Snyder Meichart families, the families I've married into. You guys are just so amazing, and it's been so great having local families nearby. You guys have just welcomed me with open arms, and I just feel so cared for and just loved by you guys, and I appreciate it so much. And it's so great to see so many of you here today. I want to thank Maria and Sean Weidman. You guys have known me almost since I was born. You guys have always been like second parents to me, which I appreciate so much. And that's been particularly true the last several years, given that you guys are based in California and I wasn't able to go back and see my family. So going and seeing you always felt like home away from home. Also, a lot of my work was done in Monterey, as you've seen. And so I went down there a ton, and you always opened your house to me, which was really helpful for my science, given that meant really cheap housing. And as you can also see, our dogs became great friends, which is really great. Which is also the transition to just having lots of cute animal photos. My cats, George and Cedric are Princess. All of you guys have probably heard me talk about them endlessly, but these brothers, which I got partway through my dissertation, are adorable and so snuggly and cuddly, and have just brought so much kind of happiness and silliness with their crazy kind of playing, and George's weird dog tendencies and other things, and Princess's strange idiosyncratic princess tendencies. But they're both adorable, and I appreciate them so much. Wilson, our puppy. Josh and I got Wilson just over a year ago. And given that he's a golden retriever, he is quite needy, but also quite lovable, which has actually been really nice, especially the last year of my dissertation, given that writing is very stressful. And when you're just very stressed writing, it's always great to look over and see a crazy puppy dog doing something silly or really sad puppy eyes guilt tripping you to go take him on a walk. And lastly, I want to thank Josh. Meeting you partway through my PhD has been one of the best things that have ever happened to me. You've definitely helped me grow as a person so much, but you've also helped my science. You're such a calm person, and you've made me much more better at managing stress and have a better work-life balance. And I appreciate that so much. You make me much more focused when I am at work, and much more relaxed and willing to go do fun things when I'm not at work. You're also an amazing field assistant, and you're always up for getting up at 4 in the morning to go help me with field work, and I really appreciate that as well. So yeah, let's go party in the Southbound. Yeah.
Okay, thank you to everyone on Zoom for attending. It was really great to have you here. Take care.